applied to the DNA, which is replication and other things. So today we have a very nice topic, which is about the chromosomal anomalies. This topic can be looked, as you see it here, as a part of molecules, because we are going to discuss how these problems will occur at the start, and also they can be seen as part of medical practice, because the conditions which are caused by these diseases or these anomalies will be reflected on the babies. So we have, for example, some congenital anomalies in which the reason for it is due to chromosomal anomalies. So today we start with certain topics concerning the structure of the chromosome. You will be learning a lot of terms. This is very important for you, because in the next future you are going to see papers or check some resources. So without these terms, you won't understand it. So try to understand these terms, which are mostly they are new to you. So these are the learning outcomes. We have different learning outcomes. We come to the structure, we come to the chromosomal basis of sex differentiation and others. Just try to make the screen bigger. This here we have. So the first few slides contain the try to be quiet. Your number is too much. If anyone just whispers, you cannot understand anything. So try to be quiet so that the lecture will be going smooth and without any interruption. So these are the learning objectives. If you read the lecture later, you come to this, you understand what are the points you have got from the lecture and not. So these are the learning objectives. Later, after you understand or you cover all the lecture, you understand these things. Now to start with, this is the structure of the DNA. This is a molecular structure, we call it the double helix DNA. So we have different dimensions. Here we have the dimension between the two DNA molecules which run anti-parallel. It's around 2 nanometers. They run in a helix from the below to the top, to the right side, and they make different topographical phenomena. For example, here we have major groove, minor groove. I am sure that in the topics of Dr. Hajin, I've covered these topics in the topic of the DNA. So we are not going to repeat that, but we are proceed from this structure until we reach the structure of the chromosome. Now, the chromosome is very practical in the process of the cell division. In the nucleus, you have the DNA molecule. We are talking now, our topics mostly is on chromosomal anomalies in eukaryotic organism. So we are discussing the human, in which we have multiple chromosomes. So this lecture will be applied to that. Now imagine you have this ribbon. It is present in this hole. And each time we are wanting to make it 2 pieces. In another word, we are trying to make what we call the cell division. In the cell division, the DNA molecule will be divided in a semi-conservative division into two parts. So it becomes very difficult, because a lot of these DNA materials will be entangled with each other. They will be entangled with each other. So what is okay is that in the different cell stage of the division, we have anaphase, telophase, metaphase, and so prophase. What is okay is that these molecules will undergo condensation. So this condensation make it a chromosome, something like this. It is very practical for its division. What we have here today, we start with this area. I'll just magnify this area. We start with this side, or this side, this side. So here we have the double helix, and we go to the left, and we have very small structure. We have 2 nanometer in diameter. Then this will be rewinded or convoluted upon each other until they will come to the level when we have the nucleosomes. These are proteins, histones, in which the DNA materials will be wounded upon them. So as you see here, nucleosomes core of 8 histone molecules, and the DNA molecule wound around them, and later they will be fixed by 1 molecule of histone 1, as you see here, so that this thing will become more applicable to be 1 unit. So here we have the nucleosomes. Then the nucleosomes, again, can see they are organizing in another level, a more complex level. 5 of them, they will form what we call it this structure, which is around 30 nanometers. So you can see all these things are going to be convoluted on each other. Then this 30 nanometers, again, if you look to the left, they will going to be also more complex in scaffolding, something like scaffold, opuscalemina scaffold, it is like that. So we have 30 or 300 nanometers, and this again will be more convoluted and more compact until we reach a level of 700 nanometers, and then if you open that, they will come to the level of the chromosome. So here we have the chromosome. It is usually presented like this, but this is not the actual chromosome. We usually represent that, I'll do that, we represent the chromosome like that. Why? Because we see it best at this stage, which is the stage before the division of the chromosome. So here, the chromosome consists of 2 chromatids. We will come to that again. But actually, the chromosome, this is a double chromosome. There's 2 chromosomes before division. So it's just before the cell division. So first, the chromosomal basis of sex determination. Here we have a healthy individual has 23 pairs. 23 pairs. Each pair, we call it a set. So we have 2 sets. Each set is 23 chromosomes. We are talking about human. Now each of them, or each of this consists of 22 chromosomes. We call it the autosome, in which it contains information regarding the body of the sexual part of that human. And we have a sex chromosome. So healthy males have 1 X and 1 Y chromosome. So we have 2 sets, while healthy females have 2 X chromosomes. We present them like that with capital letters. So this is X and Y represent the sex determination of the chromosomes. معناه شكل من كان من كان شوية من ركتان شو يحزوا كرمناز هم دايما يجسين يكلم لا بانك الموت سنزان لجيرة وقاق لجيرة وشكل أخبار ويلا means شكل جيرة ويلا and before the cell division have certain shapes under the microscope. Now each part of the chromosome has its name. Here we have a very nice of the chromosome before the division. So we have 2 parts, each of them we call it a chromatid. So we have 2 chromatids, the right and the left, you see it. If you come to the names, the upper part or the end of the chromosomes, we call it the telomeres, telomeres. While the center part, we call it the centromere. So these are, each of them, they are very, later in the molecular medicine, you will study that, each of them, they have different functions. Now, look to this area and that area. This area, it is a light area. It is composed of less tight chromatins or less tight DNA materials. But when the chromatin material become more condensed, it will make more dark area. This is, we call it the heterochromatin. So euchromatin denotes whether the DNA molecules are tightly bounded together or not. So you can see it here. Another determination, we have this chromosome, the upper part of it is smaller than the lower part. So this is, we call it the P arm, while the long arm, we call it the Q arm. So these names, you should know it, because without these names, you are not going to further understand any other things. And this is the actual picture of the chromosome. This is the depiction, and this is the actual chromosome. It is just before division. So this is two chromosomes. They have been replicated, but not divided yet. Replicated, but not divided yet. So the chromosomes are numbered and grouped. I'm trying to magnify this. Are numbered and grouped according to the size and the position of the centromere. So according to this area, we don't name them, although we have names from 1 to 22. We call them autosomes, while the last one we call the sex chromosome. So chromosomes are numbered, we'll repeat that, and grouped according to the size and the position of their centromere. So chromosome 1 is the largest chromosome, and chromosome 22 is the smallest. So against the numbers, the number 1 is the largest, and number 22 is the smallest. And usually we put the picture in the picture of the chromosome, the largest one and the first one to the upper left. We have a picture. So here we have, this is how we usually make them or rank them in chromosomes. The chromosomes times according to the position of the centromere from the left to the right. Here we have 4 types of chromosomes. We have telomeric, which is or stereocentric here. The centromere is present on the upper part, and this is not present in human, just in other animals. We have, this is acrocentric. Look to this gray centromere. The gray centromere, it is here at the peripheral part. Here they come to the middle. Submetacentric, it means it is near the center. While the metacentric, it means it is in the center. So here we have 4 terms which denotes or describe the shape of the chromosomes according to the area where these centromeres are present. Now, to investigate chromosome dividing, mitotic cells or meiotic cells are used. Cells are collected from tissue, for example, blood or bone marrow, or the fetal cells, amniotic fluid, or chronic villus, we'll come to that, and they are cultured in vitro. In vitro means outside the cell or outside the biological system. In the lab, we grow them outside the biological system. We try to let the cells multiply and we stop them. So the cells are then arrested when most cells are in the late prophase. Just before the chromosomes will divide, we stop these cells by adding certain chemicals. So the cell stops dividing, and then usually they are applied certain dyes that can be used to see these chromosomes, and later we photograph them and make them. So this is the procedure just in short. This is drawing 10 to 20 ml of blood. Other samples can be used. So somatic cells, you grow them in a tissue culture like this. Then this is some material to enhance the growth. They stop the cells, and then we put it on a stained slide like this under the microscope. We look inside the nucleus and photograph inside the nucleus. So this is the photograph inside the nucleus. It's not clear here, but what it is that you get, or you have a picture like this. This is the chromosomes before they divide. So they are mixed together like this. How we organize them? Usually with a software, or in the early days, they use a scissor and they cut the micrograph like that. And they put the chromosomes together in a usually nice manner. As you see, upper left, we start with number 1, the largest chromosome, and we come down until we reach the corner on the right, the bottom right, and this is where we put the 6 chromosomes. So we looked at these. This will show you now the numbers. They are normal or not normal, or there is extra or less chromosomes. So this process, we call it the karyotyping. We have different types. Karyo means nucleus. Typing, it is mean visualization or classification or something like this. So this is the karyotyping procedure. It is used to check the chromosomal abnormalities like this. And as you see, this is one method by which we can do that. Now, probably one might ask, what are the stains we are using? So the picture you saw is a black and white, and we have different shades of gray. This is, we call it G-band. G-banding. So G-banding, and we have also another method, which is we call it the FISH, which stands from fluorescent in situ hybridization. It uses color materials or dyes in which they will fluorescence when you apply certain wave of light. So this is the procedure of karyotyping, how you check the numbers of the chromosome whether this baby or this animal has normal numbers or abnormal numbers. So here we have the G-bands. So G-banding, it is black and white shades. So dye give chromosome strict appearance. Because it's stained regions, DNA that are rich in adenine and thymine base pairs. So here, this is the G-bands. Or the more advanced form, which can be done now, we call it the spectral. بطيفي بالشويج الزور مصر مكان. So they apply certain materials or dyes into certain DNA material that will attach to these areas that will make these chromosomes specifically, each number with a specific dye. So this is more advanced. We call it spectral karyotyping. So we have different color spectrum which are used, and as we say, we have put the chromosomes from the left to the right and from the upper part to the lower part. Now, we understand how first we check the chromosomes, we should do karyotyping. Without it, we cannot understand that this patient has abnormality or not. So you should take a picture of the chromosomes. Then you analyze that. Now, most of the abnormalities can be classified into either numerical, from their name, there is abnormality in the numbers. Either more or less chromosomes are present. You will see it here. A number of chromosomes other than 46. Any number other than 46 we call it the chromosomal abnormality numerical. Also, we have structural, we have physical change, one or more chromosomes. And we have diseases, the number are quite good, no problem with the number, but the position of the chromosomes are different. So there is change in the structure of the chromosome. So numerical and structural abnormalities, the number of chromosomes in a somatic cell is not 46, or it is not 2 sets. And here we have a very nice table, the type of anomaly. We have a group or a condition, we call it polyploidy. Polyploidy means more than 2 sets or less than 2 sets. It is not number here, it's set. So we have, if it is triploidy, we have 3 sets of chromosomes. We have conditions in which we have 4 chromosome sets. بعدين 2 set ما بينها 6 set ما بينها 12 set ما. This is we call the polyploidy. This is change in the number. We have also aneuploidy. We have definition of its name is an extra or missing chromosome. Either one is more or less. This is, we call it aneuploidy. So we have conditions, we have monosomy, which is one extra chromosome, or we have trisomy, we have 3 chromosomes are present. So here it is 1 less, here it is more than 2, or it is 3. With that, so aneuploidy, we have an extra or missing chromosome. For example, if you have in the chromosome number 21, you have 1 chromosome, we call it monosomy. If you have 3, we call it trisomy. So this is what we call it. This is the example of numerical chromosomes aberrations or abnormalities. Look here how these numbers means. The first number means the numbers of the chromosomes. So here we have 92, which is abnormal. We have 46 chromosomes. Now, after that, we add a comma and then X, X, Y, Y. This is the entity of the sex chromosomes because it may be different in males and females. So this is, we call it the tetraploidy, 4 sets. The second one is triploidy. It is 3 sets of the chromosome are present. And in these cases, we have extra chromosomes. So you can see all these. It is 47, so there is 1 more, Ecclesiastes. While in the lower part here, we have 1 less chromosome. Here also we have a different variant of Klinefelter syndrome, in which it is also an euploidy. We call it an euploidy, but it is more than 1 chromosome in number. Now the most drastic upset in the chromosome number is the entire extra set. So this is one condition. A cell with an extra set chromosome is called a polyploid. 2/3 of all triploids result from fertilization of oocyte by 2 sperm. Somebody will ask, why 3 sets or why 4 sets? So here we have one oocyte, one egg by 2 sperms. So we got 3 sets of the chromosomes. The other cases arise from the formation of diploid gametes, such as when the normal upward sperms fertilize a diploid oocyte. So here we have diploid oocyte, but 1 sperm. The triploid account for 70% of spontaneous abortion. Abortion يعني منالا باربون، منالا بارشون. So this is called abortion, and there is some condition called spontaneous. Without any reason, the baby will be coming out at the early pregnancy. So these babies are usually abnormal. So here we have 70%. Very rarely an infant survive as long as a few days with defect in nearly all organs. So lower genetic material, it is not good, it is abnormal. So the babies will die usually. Look here, somebody may, if you look at just see a normal thing, but look for number 1 we have 3, for number 2 we have 3. So this will go, so this is the triploid. 3 sets of chromosomes are present. So this is an abnormal condition. You can see it also here we have X, X, Y. So this is a result from the triploidy by certain mechanisms. Later we will check that fact. So polyploidy, which is here, it is triploidy, detected at aminocentesis. The aminocentesis, it is a procedure by which we aspirate or we take out the amniotic fluid, the fluid which surrounds the fetus. Now the aneuploidy, which is usually arrives from the failure of a pair chromosome or sister chromatid to disjoin during the anaphase. So here we have during the cell division, 1 chromosomes will not disjoin. Deko مساوي جانبه وأبيته هو يرويها تشي aneuploidy بين وتشمان جورمان. Alternative aneuploidy may be to delay movement of a chromosome at the anaphase. Thus, by either these mechanisms, 2 cells are produced, one with extra copy of chromosome, which is we call it a trisomy, and the other will become monosomy. So this is, I have put just 2 pictures just for you to understand. You go to the picture here, this is a normal one. For example, for the chromosomes. So this is spermatozoa. And this is the fertilization. What is okay, this is for male, this is for female, and this is the normal thing. But in these cases, what is okay is that look at this stage, this step, which is we call it non-disjunction. So it is supposed that the X will go to one side, the Y go to the other side during the division, but there will be abnormality. This is called non-disjunction. The X and Y go to one area and the other one become empty. So this is for the second chromosome. And this, each of them, they will give 1 sperm, spermicane, and spermanic chitiania, and chromosomic chitiania. This is we talk about the sex chromosome. So when they fertilize the eggs, it will result in conditions such as Klinefelter and the Turner syndrome. Because some of them they are empty, some of them they contain 2 sex chromosomes. So depending on the area of the non-disjunction, so this is the disjunction, they will appear in a later stage. This is also a disjunction which is occurring at this stage. So X, so one X, the other one X, so they will divide, they will give to different conditions. And in these cases, usually they give a mosaic form. It means different cells have different counts of chromosomes. خمس يلا كانوا يكنيا mosaic يعني جاجايا تكلام. So here we have the conditions, a picture which is the same but from a different source. Okay, so as you said, the triploidy is usually incompatible with life. هنالك نامت لما نكبت جوربت triploidemia and tetraploidemia, as you said, either by natural spontaneous impulsion or they will die a few days after delivery. But the other one, which is the monosomy and trisomy and some others, usually they may grow and they come out with this problem, which is the congenital anomalies. So here we discussed 5 or 6 examples of diseases in which we have abnormalities in the number of the chromosomes, either one extra or less chromosome. And these conditions, they have usually mental retardation and other problems, cardiac problem. We will have the examples, we come to the examples now. So, autosomal aneuploidy, first we have Down syndrome, which is, we call it trisomy 21. So we have 3 copies of chromosome number 21. And we have Patu syndrome, which is trisomy number 13. And we have the Edwards syndrome, which is trisomy number 18. So how they know this? They take out the samples of these patients and analyze data about مش كان بيقرأ ساكن ما بيجان جوبكن حصليها تونسني جوبكن باتيكن أما فرصة اللي سؤال كان شتيبكن شنو دروية المجال تنابي قطعا خانو إلا بخوب يخيم تو إلا يبسن. So try to be quiet and understand what I'll say. So this table shows the incidence of these conditions. We have the 2 Edward and Down syndromes. Now these incidences are not a fixed number. You might find here also, here we have this example. Some textbooks, they will give you certain numbers, they are different in the others. So this is not important. Important for you to know what is the problem behind this. So here we have number 13, number 18, number 21, and this is the percent of conception that survive 1 year after birth. You can see the Down syndrome, they will survive. The Down syndrome, they will survive usually for many years. So the trisomy or Down syndrome is most common. They occur about 1 in 800 live birth. As you see, this number is not fixed. And it is the leading cause of childhood mental retardation and heart defects. Affected individuals usually have a wide, flat skull. So this is morphologically, when you look to them, you can find many of these features. I'll repeat them. Flat skull, folds in the corner of the eyelids, spots on the irises. They may also have large furrowed tongues that cause the mouth to remain partially open. Physical growth, behavior, and mental development are retarded, and approximately 40% of Down syndrome have congenital heart defects. So this is this nice girl. She had Down syndrome or trisomy 21, and this is the area where you check the abnormality. So they take the cells, they do the karyotyping, they found 3 numbers of number 21. So this is Down syndrome. Trisomy 18, which is Edwards syndrome, which is 47 plus 18. When you say plus 18, meaning the chromosome number 18 is extra. أو شيء حاولين زائد 11 يعني 11 زيادة يعني زيادة. So this is the 18. So as you see here, we have For reasons still unknown, 80% of all trisomy 80 births are female. Clenched fists, تسجيل تأخير, with the second and fifth finger overlapping the third and the fourth finger. All the stuff, Jacqueline and here, these are congenital, and look here we have number 18 is an extra. Another congenital abnormality which is more severe is the Patur syndrome, which is incidence of trisomy 13 is 1 in 5,000 birth with maternal age effect. So multiple dysmorphic features, Shewangiagia, with apparent birth such as hypotelorism, which is mean closed eyes, microphthalmia, cleft lip palates, and other defects that you see here. Another monosomy is the Turner syndrome, in which usually we have the defect is one X chromosome. So they are females with one X chromosome. The normal female, we have two X chromosomes. So this is called the Turner syndrome. And this is the feature usually of this girl. So low sets ear, narrow and high arch palate. We have corruption of the aorta, abnormalities in the valve, low hairlines, webbed neck, say millican, it is webbed. شوية webbed, so it is webbed, not straight, and we have other conditions. Usually they are infertile. There is no mental retardation and other things. So this is Turner syndrome. Abnormalities in the extra chromosomes in the male, we call it the Klinefelter. This is one example. They are big guys, Tozegaurem, usually most males with extra chromosomes. Klinefelter have fertility problem, but few other syndromes. These are the problems usually. If you read the picture, you can find what are the abnormalities. So poor brain growth, small testicular size, female type pubic hair, long bones, so they are tall, and but as you see, they have these abnormalities. So these are 5 conditions, main conditions in which if the number of the chromosome in a baby or in a human is increased, we have problems. These are the minimum thing you understand in this lecture, because the feature may be more complicated. For example, you got mosaic, you got different types of Klinefelter and so on. This is the initial lectures for you to understand and proceed if you like to go with your career about the medical genetics. The other abnormality occurred due to the structural change, structural aberration. The numbers are okay, they are normal, but the numbers are normal. What are the things? The structure, pieces of the chromosome are not present or they are duplicated. So this is, we call it the structural abnormalities of the chromosome. And this is may occur either spontaneously, as you see here, due to the error in the recombination during the meiosis, or induced by environmental factors. And these are environmental factors. We have ionizing radiation chemicals, we have ultraviolet, we have viral infections.
Doctor, can I rest over
Now, the chromosome can be structurally atypical in several ways. It may have too much genetic material or too little. or a stretch of DNA that is inverted or removed and inserted into different type of chromosome. So these are the problems. Too much, too less, or the position will be changed. Now, to understand more the structure of chromosomal abnormalities, we classify them into balanced, meaning the exchange or rearrangement of genetic material does not cause any missing. So this we call it balanced. The same materials are present. This is the balanced structure. It is still abnormal. Here we have the unbalanced structure. Chromosome change causes missing or extra genetic material. So here we have these definitions. I'll give you 5 minutes just for rest. We'll start sharp after 5 minutes. The structure of abnormalities, we have these definitions. These are important for you because if you just remember the definition, you are not missing it with other definition. So we have deletion, which is part of the chromosome is missing. Duplication, it is part of the chromosome is twice or duplicated. Translocation, so 2 chromosomes join non-arms or exchange parts. So the position of certain parts of the chromosome are changed. Inversion, a segment of the chromosome is reversed. Axolotl, we have isochromosome, a chromosome with identical arms. 2 P's or 2 Q's, we have pictures of that, and the ring chromosome, which is from its name, its shape is a ring structure, it's like a bracelet. So this is just a hypothesis. I have put this one, later you will look very carefully. So A, B, C, D, E. So this is the short arm, the long arm of the chromosomes. Here we have deleted, so you can see A, B, C. D, E, F is present, or here it is not present. Inverted, A, B, C, D, E, F, inversion, D, E, F, go to F, E, D, and here we have duplication, you can see. So these are what we call it, these are very nice pictures. Just look to the picture and you will understand these 3 phenomena, which is deletion, duplication, and inversion. So these are examples of deletion. Deletion or loss of genetic material result in monosomy for this segment of the chromosome. أو parçayı tek parça, yani parça getir, فوقية monosomy because all parça. So terminal deletion involves a single break of the terminal part of the chromosome, and broken part are subsequently lost, while interstitial deletion in our last diagram involve 2 breaks and the inverting portions of the chromosome is lost. Here we have certain clinical syndromes. For example, deletion in the 5p, which is we call it the Craig-Chatt syndrome. Here we have Wilms tumor, Sarataneka, Toshioria Ruaya, number 15 is the PR is lost, and we have others such as rhinoblastoma and Prader-Willi syndrome. So this is the example of the terminal deletion, which is we call it Craig-Chatt. Here we have 46XX male, 5P, 5KB gamma deletion, or it is like that, female or male like that. Incidence is very low, 1 in 2,000 Earth, and this is the picture of this baby which has a mewing cry. So they call it Craig-Chatt. And also we have mental retardation and other things. This nice baby, he is abnormal. This is also abnormal. So this is interstitial deletion. We have Prader-Willi syndrome and Angelman syndrome, both of which they have missed some part of the chromosome. This is chromosome number 15. So they will lead into congenital abnormalities and obesity, as you see it here. So The duplication, you see it here. This upper picture. The lower picture, here we have the DE is duplicated. So a segment of the DNA or a segment of the chromosome will be duplicated. This is called duplication. And in these pictures, here we have inversion. So there will be some break from the left. You can see the red part, we have the upper part, so they will be breaked, inverted, and reinserted again, joined together. So this is, we call it inversion. This will also cause a problem in the gene expression and the disease. This is occur in either the short or the long arm, they can call it paracentric, while the pericentric will occur around the centromere. So the centromere is also inverted. Very nice representing picture. You can see it here. The green is upper part, the red is the lower part, so they are baked, inverted, and they will cause inversion. So these are also chromosomal abnormalities. How we can find them? Usually we can find them with fish or spectral karyotyping or other methods. There are other methods they can find these inversions. The ring chromosome, how they will be formed, usually they will be break in the broken ends, usually at the far ends, they will be broken, and they are joined together. Look at the chromosome, how it can bring shaped. So this has been seen in conditions in genetic analysis. The isochromosomes is very, just try to understand. So this is the chromosome before it divides. This is each of them, we call it chromatid. So it should divide in this plane, longitudinally. So when longitudinally they are divided, the PR and the QR are preserved. Look to the letters A, B, C, D, E, F, G. So if you break it longitudinally, part of it, one chromatid, the other are chromatid. But in this case, the division will occur in the transverse plane, correct. So when they will break, so what we have is that these are the same. So this is the form of isochromosomes. So instead of the breakage is in this plane, they will occur in this plane. So each of them have duplication and also deletion, because ABC duplicated, C or G, F, E, D is missing. So this is just trying to understand this picture. And this is an insertion, which is also very rearrangement of genetic material to non-homologous chromosomes. Non-homologous mean chromosome number 1 with chromosome number 3. Calling homologous chromosome, some your colleagues ask homologous chromosome mean chromosome number 1 from the mother is homologous to the chromosome number 1 of the father. So chromosome number 1 homologous to each year, again like if you agree homologous to father chromosome. Again father will agree homologous to chromosome. Calling men homologous mean chromosome 1 to chromosome 3, they are men homologous chromosomes. So this is insertion, they may occur from one chromosome. So this is large chromosome. They are inserted into this short chromosome. This becomes shorter, this becomes larger. So this is also a structural abnormality of the chromosome. Now this picture, I have tried to select it very carefully because it is very nice to illustrate the text. So here we have the translocation, as you see it also in black and white. So what does translocation mean? The position of certain segment has changed. For example, from this area, this piece is transferred to this one, and that piece is transferred to this one. So this is may occur, sometimes they occur between homologous or sometimes homologous chromosomes. So Also, we have different types: reciprocal, which is no loss of genetic material, and this is, you can see it here, this is reciprocal translocation between chromosome number 16 and chromosome number 22, resulted in underdevelopment and short stature child. Archer لما So this is reciprocal. This is another reciprocal in which we have the chronic myeloid leukemia. It is a clinical condition. It's caused by abnormal Philadelphia chromosome. PH1 is Philadelphia 1, stands for Philadelphia 1 chromosome that result from the balance reciprocal translocation between chromosome number 9 and 22. If you look, this is 9, this is 22, so there is some balance translocation between these two chromosomes. So these are the name of the segments in which they are translocated, and this is another, might be more rare, in which the Robertsian translocation. So two acrocentric chromosomes, they have lost break in this area, so the two short arms come here and the other one become the long arms joined together. So we have these possibilities also. Crossover, it is just a procedure normally occur during the meiosis or during the meiosis, during the formation of procedure. So homologous chromosome, like make it like this. So this is father, this is mother chromosome. They will occur is that pieces of that they will change. This is not regarded as translocation. So it is a normal procedure, a combination which is occurring in the meiotic cells. The last or the other topic is how to comprehend and apply chromosomal nomenclature, which is how we name the chromosomes or discuss its abnormality. So the karyotype formula starts with the total number of the chromosome of the cell, followed by the comma, then the X chromosomes, and then the Y chromosome. For example, normal male is 46XX, and the other one is 46XY. So this is mean a medical understandable term, how we express that chromosomes like this. And since you just XX. Other notations or other terms that are used following this, each time separated by a comma. The plus or minus signs followed by a number indicate the extra or missing entire chromosome. So from this example, you might understand it. Now, when a piece of the chromosome is missing or a chromosome number is indicated, followed by a P or Q, a minus or plus, mean that the missing arm is either P or Q. Other notations, heterogyny missing minus P5, mean there is loss of the P5 and so on. Or sometimes we can write something like this: deletion, duplication, inversion, satellite, translocation. Now, don't expect yourself to understand all the terms here. I by myself, I have graduated more than 30 years ago, still reading about them. The idea is that medical genetics is a specialty. Specialty of so it is a specialty. There are too much information. So we try to give you just a few things for you to start with and understand your topics. So here we have the table. I have put that like this. This is the numbers, how it is expressed, and this is the problem with them. So later you can see, for example, 47X YY cis Jacob syndrome, which is a male with 2 X chromosomes.
Why?
Oh my word, negative. The other topics, first we have referral of patients for karyotyping. Somebody asked, what are the patients, how you select? So first, indication of routine chromosomal investigation. First, constitutional or congenital abnormalities. مناطق لديك اشتكي هي جاري هي تلفريكي هي اشتكي هي داون syndrome يعني اشتكي دكتور pediatric exam. So this is one condition. You take some cells of this newborn baby and you try to analyze the difference if there is a problem in the chromosomes. A prenatal screening for Down syndromes, especially raised in maternal age, agar diet disorder or family history of chromosomal abnormality or abnormal ultrasound. أنا حمي والي إلا بدأ يكونوا ماشي دواء وأنا كف حسبين. Second, birth defects, malformation, mental retardation, if it occur in the families. Abnormal sexual development such as Klinefelter, which is abnormal, not a quite right male. Infertility or recurrent fetal loss. So try to find the causes by forming this investigation. Also, they can be acquired abnormalities such as leukemia or some related conditions. We have some condition in which genetic abnormalities, that's one thing. We'll see in North Philadelphia and the myeloid leukemia and so on. So the diagnosis of chromosomal abnormalities enable first accurate diagnosis. You diagnose, you know the condition, and also you know the prognosis. And we expect the future of that patient. If he is Down syndrome, we expect that he will not live more than 45 years old. If he is Edward, and this is this. So this is prognosis. So prognosis of clinical problem in the patient. Second, better management of affected patient, for example, hormone treatment in some sex chromosome abnormalities, understanding the reproductive risks of the carriers. This is genetic test for the couple, for the male or the mother and the father, future to be. So they will do that to understand if they have risk to bring a baby. So this is another option. And prenatal diagnosis of possible termination of affected pregnancies. They have abnormal baby in the first time, and the second time she becomes pregnant, they will ask if they can terminate that or not. So they will do this karyotyping. One thing about fish, I am not sure have you covered that. It is very simple. So they will attach certain material dyes into certain tags. The tags is DNA molecules, and they put that on the chromosome. It will be attached to the chromosome and specifically at that area, and they can be seen. So this is, we call it fluorescent in situ. In situ means in position hybridization. So the dye is hybridized. It's joined the DNA molecule through a DNA tag. So this is more advanced method to see the chromosome itself. So fish can identify chromosomal abnormalities, help in the gene mapping, analysis of chromosomal structures, aberration, and whole determination. So many laboratories that perform amino-synthesis or CTDS uses fish for chromosome 13, 18, 21, because these are the commonest one. And quickly identify the most common problem. معناه نحن common terrain. try to. So this is fish, specialized stain, Amycelica, fish So this is telomeres. So in this case, they use chromes for the telomere. So these chromes, the dye will attach to the telomere. So telomere glows towards the end of the DNA. Also here we have fish show 3 chromosome dots that correspond to 3 copies of chromosome. This is number 1 chromosome 2 هناك بين الديكن ورسمة كان بارشة يعني except لو رسمة أمبر هناك كان بين معي هذا كان جوانتا ماشي كان So 3 dots, each is for chromosome number 21. So this is the usefulness of the fish technique. You can understand that. So here we have translocation. أنا طبيعية أنا تشيا parcheki skoro parcheki salsa. So there is translocation. The way to So this is translocation. So using fish technique is very nice and important. And this is the fish technique showing the symptom. So we have different techniques to understand that. Okay. The other things are the genetics for detection of chromosomal abnormalities, apart from the karyotyping. So prenatal diagnosis, prenatal means before birth. Prenatal diagnosis include all aspects of embryonic and fetal diagnosis. It provides reassurance without which many would decline to undertake a pregnancy. ليش لو أنا ألتراساوند أو أفرد كريم وإبزان أو المعلقة باش نية باش بيت continuation again normally باش بيت احتمال termination. Now in practice, 93% of prenatal tests provide assurance for the couple, and selective termination of pregnancy is taken only in 7%. Prenatal diagnosis techniques may be divided into 2 groups, invasive and non-invasive. So invasive means there is some sort of invasion or some harm to the baby and the lady which is here. So this is what we call invasive. Regaji توزيع تستربنا خوشك. ولمنا من endoscopic وثمانو مع دي سلامة كمبيوتر invasive. طبعا توزيع ثمانية ولا مليان ميز توزيع ميزي ولا مليان أنا invasive مليان سكان وقي. أشتاق لسر مثلا ultrasound. The Sersky was a Julian. This is non-invasive. So here we have invasive and non-invasive. So it's a genetic test for chromosomal abnormalities. So here we have invasive. First we have amniocentesis. So these are the common. We have chorionic villus sampling and synthesis. And we have non-invasive, which is the most common, is ultrasound. Sooner كآسان كيتشناري راح نشوف الجزيرة وخاطر وهذا preparation material and other types of imaging also. So what is amniocentesis? This is the amniocentesis. So this is done in good centers. So fetus at the age of 15 or 16, usually they go amniocentesis. This is containing the amniotic fluid. So under the guide of ultrasound and other things, usually they introduce this needle and they take amniotic fluid. So this fluid contains fetal cells. So they analyze it to see if there is chromosome or chromatic or not. So this is amino synthesis, and this is new inspiration. Aspiration, synthesis. This is very nice ultrasound. This is chorionic villus sampling. They aspirate various from this chorionic villus. This is the abdomen through the vagina, so you will use this device to this area, take some parts of the villus. These are structured physiology like one and analyze it. So the most common is these three amino synthesis and the chorionic villus sampling and ultrasound. So amino synthesis is the draw of the milk fluid is usually performed in this. And they take the fluid and examine it. Different examination can be done. And according to the result, they can inform the lady to do the pregnancy continuation or termination. Now, one of the most things which is somebody expects from a baby while he was not born is to know the sex determination. So it can be done by ultrasound. In early days, there was no ultrasound, so they might use other things. This is, we call it the determination of the sex of the baby by using a condition or a technique by finding these dots, which is, they are called bar bodies. Bar bodies. So these bar bodies, let's magnify this area. So this is the photomicrograph comparing cheek epithelial cells, cheek epithelium, هلا now دموي عند الفلوس لا يجي عند الدور عند الدور from male that failed to reveal bar body to the right with a nucleus from the female that shows a bar body. So indicated by the arrow on the left. The structure also called a sex chromatin body represent an inactivated X chromosome. Now, I need a عملية basic and so this is one method of sex determination. What are other methods in the early condition sex determination? أما طبعا الأخيرة ولا الأخيرة طبعا ما قاعد كتير يعني ما سيشيكنا karyotyping. Karyotype, you take the chromosomes and you take a picture and put the X and Y chromosome. If they have two X, say this is a normal male. If they have X and Y, say this is. So by karyotyping also we can determine, apart from that, we can also determine whether this baby is a male or female. The last slide is for you to go and understand. This is about ethical issues associated with the genetic testing. Ethical issues, ethics. ما سيجي ما شتكي راست يعني كينيان شتكي الله شيء الله بدي شيء أنا راسته جنيك نالي جنيك تولم نالي تو أخوش كيان يعني بين عليك سبيني من على كي بيتو تشتغل فمنالي شيء أخوش وقتها كم صار لك شيء بنو نالي زيادة. So these are problems that ethics that will allow us. So we have ethical and also we have religious problems. So these include the respect of privacy, autonomy for this person, the couples, or for the baby himself, personal best interest, responsibility for the genetic health of the future children, maximizing social best interest, minimizing serious social harm, and reproductive liberty of the individual. Now, one of this common one is now we have is thalassemia, which is put it like this, because each of the mother and father, they may have a triad for thalassemia. So if they bring babies, usually they produce a baby which is from the early days he become anemic, and they need all the time with him for transfusion and for different problems. So there is logic for making a baby or not. Some of them they say we like the babies, others they may not decide to bring babies. So these are some of the ethical issues. Cost effectiveness, solidarity, mutual aid, and respect of difference. So all these are important in the genetic analysis. It is like, for example, you have certain password for your accounts in the emails or in the bank accounts. So it is just concerning you. My health is concerning my health. Again, I don't like other people to know about my disease, about my conditions, and I carry an abnormal genes. So these two slides about. So there are also different kind of risk. This is about the risk involved in genetic testing. These include physical risk, the amino-synthesis, planning physical care, or we have emotional, social, and financial risks also. So in some cases, genetic testing create tension within a family because the result can reveal information. So problem will be seen early in stage. And also we have psychological risk with guilt and anxiety, impaired self-esteem, social stigma, and insurance and employment discrimination. من خارج مهم يجبزان النخوشية كده تعينية كم تعينية كم insurance كده. So this is for genetic testing, not just for chromosomal testing. Okay. Have a nice weekend. I'll be here if anyone has